Okay, students, so today's lab is about agarose gel electrophoresis. So, agarose gel is a way, or gels are a way to separate DNAs on the basis of the size. So, first thing, this is the gel counter of the lab. Okay, so what we have here is like various gel running apparatus. So, this is, for example, used for power supply, which creates the voltage for gel separation. This is for protein SDS page gel running, so protein gel separation. Also, we use it for DNA separation, small DNA separation. And on this side, we have the agarose gel apparatus. So this is the gel chamber where we actually, so this is called a horizontal gel electrophoresis, where this side is the positive, that is the negative. So we separate the DNA basis on the size using the charge. Okay, so here is the gel assembling station. So what we do here, so this little cassettes. Okay, the cassettes. So here we pour the gel and let them polymerize. Okay, and this ones are the combs which will create the holes where we will load the DNA. Okay, so Sanatan will show how to assemble a gel. Okay, so this is, he is tightening the screws so that it becomes watertight. Okay, so here he is putting the combs. Okay, so you can see very closely here. So if you pour any liquid, so it will cover the part of the comb, and then create the holes there as it polymerizes. Okay, so that is that. So this is, so we are creating two types of gel today: 0.5% and 2% agarose gels. Okay, so here we have a 1x TAE buffer, so tris acetate EDTA buffer. Okay, so we need total for 50 ml for each type of the small gel. Okay, so we have a bigger gel cassette. So this is a bigger gel cassette that is on the other side. Hard to see from here. I'll show you once this is ready. So 50 ml for each of this is required. Okay, so we'll show you the next steps. Okay, so the 250 mLs are ready. This one I just wanted to show. This is a bigger gel cassette, so this is requires 100 or 150 ml. So this is for this part where you can make, like, it has two lane positions. So you can see these are the two lane positions. So we can show. Okay, so this is one, and this is one. So these are, each of these are 24 lanes. Okay, so that way you can make a lot of gels, a lot of positions at a time. So we'll now show how we make the gels. Okay, so here in the weighing machine we are weighing the agarose. Okay, so this is the agarose. So you can see that this is a typical agarose. We use it for separating DNA. So this is low melting temperature. Low EO means the size of the pore is small, and whatever reagent we use, that is must be for molecular biology. Okay. Okay, so tearing right now. So here Santan is now weighing the agarose. So I'll let you calculate how much you need for 0.5% and 2%. Okay, in the students. So he is now weighing the quantity. Okay, so 2% is there. So it has not dissolved. So if you look at this forms suspension. So we have to microwave it to dissolve it. Okay, so again not dissolving completely. Okay, so it's a suspension. So we are going to microwave it in traditional microwave. So requires about 3 minutes at least for um, for this to completely melt. So once it melts, it becomes homogeneously and clear, transparent. But then you have to pour it quickly so that before it repolymerizes again. So we'll wait till it is done. So this is after 3 minutes, and you can see that it is completely transparent. Okay, so it is completely transparent. Okay, so both of them are completely transparent. So now, thing is that we have to put ethylene bromide, but not right away. So basically, one has to wait at least till this temperature reaches around water. So until the temperature reaches at least around 50 to 60 degrees Celsius. How do you know that? So you'll be able to touch it with your hands. Okay, so now you can add it is around 100 degree. Okay, so it will take about 20 minutes to reach to about 50 to 60 degree. So once it reaches that temperature, you add the ethylene bromide. Okay, so ethylene bromide is the staining reagent for the DNA. Okay, so we'll see after 15 to 20 minutes.